Hello everyone and welcome back to PM1A. Today I would like to introduce you to the Genes and Chromosomes lecture series, also referred to as genetics. Within this lecture series we will be having a short introduction, that's the session today, followed by fundamentals of nucleic acids. In this session we will learn how DNA and RNA look like and how those molecules can actually store genetic information. In the following, we will learn how DNA is generating identical copy of itself, how it replicates. We will also have a look at the process of transcription, where the information stored at DNA is transcribed into RNA. We will also learn how the genetic code works and how the information on the RNA is finally translated into fully functional proteins. After those 4 sessions, we will have an exam preparation session dealing with the content of those lectures. For those exam preparation sessions, it is really important that you guys ask questions. If you struggle with certain lecture, or if you would like us to explain a certain lecture in more detail from a different angle, please do ask us using the dedicated Padlet or via email. In addition to answering your questions, we will also go through several multiple choice questions in those sessions, and also talk about short answer questions dedicated to the content. After these preparation sessions, we will learn how DNA is organized into chromosomes and how gene expression is regulated at different levels in our cells. In addition to this, we will learn about mitosis and meiosis, which are both processes how our cells divide. They are similar, but you will see in the dedicated lectures that those processes do have quite a few differences. The last session within the genetics lecture series will deal with a very interesting and modern aspect of genetics. We will be talking about gene therapy. And gene therapy is very present in the media, and I will show you several examples how genetic can be actually applied to treat and cure patients suffering from disorders that were previously without cure. After those lectures we will have an additional second exam preparation session, so also here please do ask questions. If we think about genetics and DNA, then we could also think about diversity. We are all different, and so are our cells. Our cells come in many different shapes, and they do fulfill very different functions. We have bone cells that provide us with stability, we have neurons that are crucial elements of the central nervous system, we have retina cells that are important in our vision. As you can see here, those cells do look slightly different, and also they do have very different functions. Some of those cells are even able to move, like sperm cells. However, the genetic information carried by all of those cells is mostly the same, and it is contained in complex molecules called deoxyribonucleic acids, short DNAs. DNA is usually stored within the nucleus, and the nucleus is gonna be a topic of one of our cell biology lectures, but for now please do keep in mind that the chromosomes are stored within the nucleus. In addition to the nucleus, some DNA is actually within the mitochondria, but we will learn about those organelles in a dedicated lecture too. So, what is genetics? If we look up the definition, then we will find something like this. In this definition, Genetics is the study of heredity and the variation of inherited characteristics. Another definition is that genetics is the study of genetic properties or features of an organism, its characteristics, and so on. Fine. Sounds quite fancy, sounds important, but why is this important for you as future pharmacists? And in order to highlight to you why pharmacy is actually relevant not just for the development of pharmaceutics, but also for pharmacy practice, I would like to share with you a short video.
UBC and 20 pharmacies across the province have joined forces for research. The Shoppers Drug Mart in Pine Centre Mall is one of 3 pharmacies in town taking part in the research, which looks at the genetic interactions between patients and their prescriptions. The testing will involve taking the saliva from those patients prescribed the drug warfarin. The aim is to better personalize prescriptions to better outcomes, and here is just one example.
So a lot of people know that codeine either works well for them, or it doesn't seem to work at all, or it works okay but not so great. And the reality is it's due to your genetics, because codeine gets metabolized into morphine in your body through an enzyme in your liver. Depending on how many copies of that enzyme you have will depend on how much morphine you make and what your response to the codeine is. So this is most tragically illustrated in certain infants who became codeine in the past. Unknowingly, they were rapid metabolizers, so they produced a large amount of morphine and went into respiratory distress.
So, as you've seen here, genetics is obviously more and more important in daily pharmacy business, because many pharmaceutical companies are nowadays developing personalized or precision medicines, and I'll be teaching you more about precision medicine and personalized medicine in year 3. In addition to this, a very modern application of genetics is gene therapy, and gene therapy is very promising because it uses genes to prevent or treat disorders. And in future, this technique may allow us to treat disorder by inserting a gene into patient cells instead of using drugs or surgery. In addition to this, if we are talking about genetic disorders, we could try to repair the gene defects in the cells of the patient, thereby curing them. In this approach, we would correct the faulty copy of a gene, expand the cells of the patient in a dish, and then put those cells with the correct variant of the gene back to the patient. And in order to achieve this, we need several tools within that you will learn within this lecture series. In addition to gene therapy, genetics is extremely important for so-called pharmacogenetics, and pharmacogenetics can be simply defined as the application of genetic analysis to predict drug response, its efficacy, and also toxicity, because for every given drug we might have different population of patients. We might have patients that are able to metabolize the drug very fast, very rapidly. We could have the population of patients that metabolize the drug normally, and we could also have patients that metabolize this drug slowly. In this case, we would have to adapt the dose we'll be prescribing based on the genetic predisposition of the patient. Overall, we can use pharmacogenetics for drug discovery and development as well, because we could identify potential responders and non-responders to a drug at a very, very early stage, saving money. We can also use it for prescribing. We could, in theory, perform genetic tests and use it as an efficient factor in determining the best choice of drug for a given patient and condition based on individual genotype. And this type of testing is more and more common. So why do we have patients that are able to metabolize drug fast, slow, or normally? As you can see here, we can have very different causes. We can have certain gene variants in a patient that are actually causative for those different rates of metabolism. In this table you can see few examples of genes that affect metabolism of drugs quite severely. And also it is quite evident that many different processes within a cell can be affected by genetics. Genetics can affect pharmacodynamics, it can affect pharmacokinetics, it can also affect cell metabolism. So all of this information can be used in pharmacogenetics quite efficiently. In addition to this, rather applied genetic testing is worth mentioning that genetic testing is widely available right now, and it's also quite cheap. As you can see here, you can buy on Amazon a genetic testing kit sold by a company called 23andMe, and this test can tell you quite a lot about certain risk factors that you might be carrying in your genes. As a future pharmacist, you should be able to advise patients. You might face a patient that would like to buy such a test on Amazon, and the patient could have realized that he has an increased risk of developing Parkinson's disease. So you need to be able to explain the patient what this result actually means. So those are all applications of genetics. But what happens when genes go wrong? Genetic mutations can have a lot of different consequences. A very classic example of a genetic disorder is PKU. Another disorder that is less common but still can be found within the general population is neurofibromatosis, and I will show you how it looks like later on. We have disorders like epidermolysis bullosa, and that's a disease of the skin where children affected develop chronic injuries, chronic inflammation and chronic wounds on their skin. There is actually a quite large number of genetic disorders, and if you're interested in this, I recommend that you have a look at this list of genetic disorders on Wikipedia. The first example I would like to show you today is PKU. PKU is a rare genetic condition that is present from the birth. It is inherited, and in this disorder the body is unable to break down a substance called phenylalanine. This builds up in the blood and brain, and in very severe cases this can lead to brain damage. Usually PKU cannot be cured using traditional means. However, we can, to some degree at least, manage the symptoms. So we can apply low protein diet, which reduces the level of phenylalanine in the body. So if the treatment starts early, patients can actually be quite lucky and don't develop any symptoms. But if it isn't recognized and treated, the damage to the brain can be really severe and result in learning disabilities, behavioral difficulties, and even epilepsy. Another example of genetic disorder is neurofibromatosis 1, and this is the name for a genetic condition that causes tumors that grow along peripheral nerves. In this disorder, a mechanism in the cell which negatively regulates cell division is affected. Therefore, the cells divide in an uncontrolled manner and form those tumors. Unfortunately, currently there is no traditional cure for neurofibromatosis, Both there is light on the horizon, as you will see in a lecture dedicated to gene therapy. The final example I would like to show you today, and this is a disorder I'm really passionate about, is epidermolysis bullosa. This is a very severe condition of the skin, and as you can see here, children suffering from epidermolysis bullosa develop chronic wounds in their skin, and they suffer from chronic pain and inflammation. So this is a very serious condition, and unfortunately, apart from wound care and wound management, there is no traditional treatment option. Also here, luckily for the patients, gene therapy shows a lot of promise, and indeed first patients have been cured using gene therapy last year. And again, we will have a closer look at genetic therapy for EB in a dedicated lecture later this term. So, what is the recommended reading before you engage with other online lectures within this lecture series? I recommend that you read the newsletter provided by the National Institute of Health in the USA, and I will post this material in the lecture folder as well. You should also read about the uses for genetics in pharmacy, and an article explaining why pharmacists begin to embrace pharmacogenetics. With this, I would like to close this session, and I'll see you soon in our following lectures.